So maybe some of you have already started in collecting data in the field. Maybe you're trying to create your methods, but most likely you have no idea what exactly you're supposed to collect. And hopefully this PowerPoint presentation here will help kind of ease some of your concerns and give you more clarification on this issue. And if it doesn't, please contact me and we'll find out a way for you to collect data in which it will, in a way, answer your hypotheses. First, before you get out in the field, you need to ask yourself, is the data that you are going to collect actually answer the question that you first imposed? And this seems very simple, but a lot of times individuals will think the data that they're going to collect or they want to collect will address their question, and after they collect it, they go into the computer lab to analyze it and write it up, and they realize that they've collected a totally different type of data set than they initially wanted to. So things that you need to think about is, remember last week we talked about response and independent and dependent variables. So by measuring your response variable or your dependent variable, will that link back to your independent variable? Are the two linked? So if you're trying to measure a change that occurs, maybe up a gradient, is what you're measuring actually going to address that change? And then also, what is the type of data that you are going to measure? Like what are the units going to be in? Is it going to be in plants per square meter, percent cover, moisture perhaps? You need to think about that. Then also, randomization is a big aspect of the scientific method. We went over this in a PowerPoint, I believe, in week 3 or so. And you need to randomize your plots or transects or however you choose to collect your data. And we can talk a little bit more about that if need be, but the whole idea is maybe you have your site, you create a grid of 100 specific sections in which you could put down a plot, and you pick out of a hat. You have 1 to 100 in there. You pick out of a hat, maybe you say number 2, and that's the plot location that you had labeled a 2 in your grid. Then also sample size. You want to make sure that you collect enough samples to represent your entire site or address the question that you were trying to answer. And here I like to always live by the rule of 10. I think 10 is a very good minimum number, but also you want to balance this in the detail of your sampling with a number of samples. So I don't want you out there, every day and every night for 2 weeks, but I want your sample regime or your sample sizes to be enough to show me that this represents your specific site. Then also data that's collected, in this case, I want to be quantitative, so it's got to have numbers. And for an example here, like if you're measuring water depth, 15.5 millimeters, and then there are 18 Phalaris arundinaceae, which is a plant species, which I think I got that name wrong. But either way, reed canary grass, or 1.56, which is diversity for a plot, right? And here you want everything to be metric. So metric is the accepted kind of numerical system for scientific papers, because we want the rest of the world to read these papers as well as learn something from them. Before you get out into the field, you should and you need to create a data sheet. So in this sense, you want to be able to put a lot of details about the data that you're collecting, such as the date, the time, as well as the weather conditions, the location. Please attempt to use geographic coordinates on this, and any observations that may exist. This could be an example of, you could see deer tracks that are going through your site, there are kids playing, there are some cigarette butts which show human disturbance and use, and anything that you think would affect the presence of plant species or whatever data you're trying to collect. And in this example, using percent cover, and I have the plant species that are within my plots, and this is a form of transect. So if you're doing a transect, and we'll talk about this in upcoming slides, but if you're doing a transect, you want to mark the transect number, as well as this can represent the different quadrats or subplots of this transect. And then you would put either the abundance, the number of these kind of species there, or the percent cover. What is the total area covered by these individuals? and attempt to organize your data initially so you can easily plug it into an Excel spreadsheet and get the averages, be able to run t-tests, be able to make a nice graph, whatever you think would help make your life easier. If you think about it ahead of time, you'll save yourself hours later. So the two main types of data collection which I think can be applied in this class is the form of transects and plots. So this is a way to gather vegetative data within an area. And here this plot design right here measures the kind of efficacy of herbicides. So it's looking at comparing these specific plots in which they sprayed herbicide or they didn't, or maybe they did seeding as well. But as you can see, there's a buffer, and this is more experimental, but this is kind of the setup that you want to be able to generate for your study. Transects. So why transects are important is the fact that they track changes or that dependent variable, what you're measuring, with a distance. So you can see how a plant life will actually change, and this is a height as you go over a mountain range. Again, this is out of the scope of what you're doing, but it can definitely provide a good insight of what a transect is and why it's useful. So here they mapped elevation, and then also they mapped the plant height of a specific plant as well as in its life cycle too. So they showed like the morphological differences that occur with this one plant as you go up in elevation or eastward across a mountain range. I think this is very interesting. And the height in this case is the dependent variable. It's what they're measuring for, and the distance along the track, which represents elevation, is the independent variable. So that's how you can make that connection. In your case, you could be doing this as slope, distance from water, the further you enter into a wooded environment, how does the understory change, or possibly away from a path. The distance away from a path, how does a plant community change? So at a closer look here, transects can be seen as systematic data collection along a specific line. A transect is simply one long line that you follow, and this is appropriate when you're measuring a variable along a specific distance. So again, if you're changing in slope or water usage or percent tree cover gradually, you can measure how a specific variable changes along that gradient as well. And there's two different ways in which you can carry out transects. You can use quadrats. The idea here is that you use a 1 meter by 1 meter small plot, you could say, which we saw in the picture. It was in the title slide, was a quadrat made out of PVC pipes. You can refer back to that. But the idea there is that you simply look into that quadrat, that plot, and you assess what is in there for abundance as well as percent cover, if that's what you're measuring. The benefits of this method is that these quadrats provide detailed sampling along a representation of an area. And these are good for small areas because they are high in detail and you do move a little bit slower, but that's what you're going for. If you have a smaller area, you like to use quadrats so you can get a full representation of the species there. And again, kind of the downside is that they're a little more time consuming and it may result in a shorter transect area that you may be carrying out. And when I kind of indicate a quadrat, this means that you put that 1 meter by 1 meter subplot or quadrat like every 2 meters. So maybe you have a 20 meter transect line and you want to measure the plant diversity every 2 or 3 meters. So you're able to do that. So these boxes will be seen as our plots or our quadrats, and it goes along at a 2 meter intervals, you could say. And then you'll be able to sum those up at the end and get a representation of your entire transect. So then point intercept transects are another method in collecting data, and this is carried out in smaller intervals than compared to maybe this 2 meter break. This could be possibly 10 centimeters or 20 centimeters, but the reason why it's given the name point is because you're simply looking at the individual present at that specific point, so where that line hits, and you count one individual at a time. And the benefit for this is that it is able to cover a much larger stretch of land than your quadrat transect would. So here you could honestly do 200 meters in maybe 20 minutes if you just count the plants there along the way. And in this way, it's a great comparison. So you'd want to be able to compare the individuals in this transect compared to another. So it's not as useful for going along a gradient like the quadrat transect is, because you don't you don't break apart the distance. But you could do it for every 10 meters if you measure at 10 or 20 centimeter intervals. But this is more of a way to compare, say, an area that was highly disturbed due to human impacts to an area that was pretty intact and is currently pristine. So plots. Again, this isn't as detailed because this is very simple. You basically break down an area, and one plot would be this whole box right here, and you would count the different individuals in there. So this was taken from your week 3 dispersion slides. So this is an example utilizing plots. And the smaller 4 plots in there are seen as subplots. So you have 4 subplots within this larger plot. And this is a great way to measure differences in 2 areas, such as how do different levels of disturbance affect plant life. Canopy cover is one out in a field is one underneath alders or aspens, as well as land use. Kind of coincides with disturbance. Is this land used as a recreational area, or is it preserved as a natural area for biodiversity conservation, etc.? Now, the types of data that you will be collecting. So abundance. Abundance can be seen as a simple count of the number of individuals present. So saying that subplot 1, maybe there were 8 garlic mustards, 3 reed canary grasses, 2 columbine, and 5 red clovers. That's an example of that. You could compare that, and by the subplots you could calculate the biodiversity and compare that to, say, another plot or subplot. And then percent cover is the total cover of species within a plot or transect point, and that's observed right here. And what percent cover means is you basically just stare down into your allocated plot or quadrat, and you look to see how much of one plant are you looking down at and how does it compare to the rest of the site. So if you look down and you see 3 patches of red clover, you would say that translates to roughly 25% of the plot. So although that seems simple, it is one of the main kind of parameters or data collected in plant ecology experiments. So although it's not rocket science, it still is science, and it's very useful, because not only does it provide, it doesn't provide the abundance, it doesn't provide the number of individuals present, but it provides their overall presence. Like, what is their biomass relative within this area in that way. So I kind of talked about biodiversity, which you can calculate through both the percent cover and the abundance. And I didn't put the equation here on purpose because I want you to refer back to your week 1 slides. But that whole idea is a ratio of individuals to the other. So you could use both abundance or biodiversity for this case. And then the Moore-Sita's index of dispersion, which we talked about in week 3, requires abundance to be taken into account for the calculations to be made. And again, this to understand dispersion patterns, you'd want to use a very large area. So in this case, possibly cover like a huge site, but you could do tree counts. You could do how trees are carried out within or dispersed within a large park. And it may take you a few hours, but the tree count can be done and can be accurately calculated with a Moore-Sita's index of dispersion. And this is just an example of a simple Excel bar graph that would be awesome if you were able to put that together. And it's just using mean percent covered between 2 treatments with